From Microbe TV, this is TWiEVO, This Week in Evolution, episode 21, recorded on July 12th, 2017. This episode of TWiEVO is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase with free shipping by going to blueapron.com/twee. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Greetings, Vincent. Great to be here on TWiEVO's 21st episode. TWiEVO is legal.
Well, let's have a drink.
That's right.
Last time I saw you, you were holding a beer and fishing. Did you ever catch anything?
Guilty as charged. Yeah, you're talking about at the ASV meeting in Madison, Wisconsin, near the shores of Lake Monona, fishing for bluegills, and came up empty. Zero fish that day. 0 bites.
I have a picture of you fishing, so it can be verified.
Yeah, it was fun. It was first of all, a great meeting in Madison as usual, and great to be back in that town. Really good energy there.
Madison's a good town. Yeah.
Yeah.
However, you disappeared after the first 2 days.
Yeah, I was a little bit of a ASV ghost. So I had a few deadlines that I was horribly behind on. And so I hung out the first day or so for we did a session on evolutionary virology. This was organized by Colin Parrish. Turned out to be spectacular. I thought great science from a bunch of folks. And then, but then I kind of had to make myself scarce to get in a few things on time. So I was there in spirit and personally in person, part of it, but not the whole way through.
You're a busy guy.
Well, looking for that balance, and it's still elusive, at least for me.
I don't know. I'm looking at this page here, and there's a quote in the front that says, "Work hard and play hard."
Yeah, that's a good idea.
We have a guest today on TWiEVO.
I know. I'm excited about this.
And he's actually a crossover from one of our other podcasts. He's from Austin, Texas. Rich Condit, welcome.
Hi, everybody. I was Skyping around looking for TWiEVO, and I got lost.
I got lost.
What's going on here?
You stumbled into TWiEVO.
Pretty weird. So
yeah.
I'll hang out. This sounds good.
Yeah.
Hi, everybody.
It's a good podcast, Nels and I.
You're calling me a crossover. Is this legal?
It's legal.
Oh, yeah.
It's all legal.
All past the 21st episode.
I just heard that TWiEVO is legal, so yeah. Okay.
21.
Indeed.
Yeah, so we started, I first talked to you 2 years ago at ASV, right, Nels?
That's right. This was when you were president of ASV.
Yeah, up in Canada, yeah.
London, Ontario. And so here we are 2 years later and 21 episodes old.
Bad, right?
Yeah, I think it's great.
I say we're building. We're still building. We got a ways to go.
Well, and we're starting to attract, top shelf guests like Rich Condit, who
That's right. He said yes. Yeah, he said yes. I don't know. Are you on TWiEVO this Friday, Rich? I forgot.
Oh yeah,
you're here.
There.
Okay.
I just read the paper this morning.
Yeah, that's an interesting. We have a guest on TWiEVO this week. Also, we have Ben Tennover.
Oh, he's great.
He's going to come up from Mount Sinai. I heard his talk at the same session at ASV that you just mentioned, Nels, the evolutionary biology session, the satellite. And
yeah, wasn't that exciting?
I said to him after, I said, "It was a great story. You got to come and tell it to us on TWiEVO." Because he put the story together so beautifully, right? I want him to sit here and do the same thing without slides, though.
Yeah, agree. I think that will be a really fun conversation.
His paper came out a week or two ago, and last week we did a RNAi paper on TWiEVO also, so we're going to have a little mini arc.
Yeah, that'll be good, because I'll be interested to get Ben's reflections on the paper that we did last week as well, and the whole RNAi in metazoans thing.
He put a very interesting tweet up the other day. He said, RNAi in mammals, whether or not it exists. Looking forward to the discussion.
Yeah, that's a spicy topic these days, controversial topic with some real strong opinions on both sides, which is always interesting.
Have you seen this recent paper, Nels, saying that Enterovirus 71 has a viral suppressor of RNAi, and they took it out, and all of a sudden the infected cells are full of small viral RNAs?
Yeah, so I saw the abstract or hints of it, but I haven't had a chance to really dig into it. But
it looked good to us.
Yeah, that's what I'll take a look under myself under the covers there.
Just listen to TWiEVO.
Exactly.
The discussion's pretty good.
Yep. I'll wait for Tennover to weigh in as well, and then I'll listen to him back to back.
You can do it. Yeah, you can do an RNAi binge.
Yeah, that's right.
Somebody said on Twitter also, I'm starting to listen to TWiEVO from episode 1, and I'm going to go through all of them in order. Has anyone ever done that? I said, I'm sure there have been.
Wow. Godspeed.
Yeah, really. Well,
how many episodes?
How many episodes of TWiEVO?
We're doing 450 next week, right?
450. Tomorrow, 2 days from now, Friday, yeah.
Very cool.
Well, Nels, we have an interesting paper today.
So wait a minute.
What's up? You need the weather?
We don't do weather. You guys not do the weather?
No, you're not here, Nels and I just charge ahead,
Okay.
Tell us your weather over there in Sun Valley.
Well, Sun River. I'm just really pleased about this. Sun River, Oregon now, it's 83 degrees and sunny. The humidity is 15%.
Ooh,
the dew point is 29 degrees. This is just amazing, amazing climate in the summer.
That sounds perfect.
We have 32 Celsius here in New York City, 51% humidity, and it's mostly cloudy. But you, Nels?
Mostly sunny. Salt Lake City, we're hanging right at 90 degrees Fahrenheit, which is actually almost a relief, given some triple-digit business for the last week or so. It's been unseasonably warm. But some nice afternoon thunderstorms kind of perking up from time to time. And actually, I'm looking out across the Salt Lake Valley right now and see some big thunderheads and some what looks like some showers coming in.
Do you have relatively low humidity in Salt Lake?
We do, and that sort of saves you on these, triple-digit days. The nights actually cool off and moderate really nicely. Yeah, exactly. Yep. Salt Lake is semi-arid, I think, is the designation. Not quite as dry as Arizona or something like that, but kind of in between.
Now we have a paper today, and that's why Rich is here, because he had. I think what was the timing on this? So I think, Nels had sent it to me a couple weeks ago. And said, let's do this. And I said, okay. And then you then emailed me, here we go. It was July 11th, not that, July 10th.
A couple of days ago.
Yeah, you wrote, this is cool to me anyway.
Mm-hmm.
Let's do it on TWiEVO. And I said, well, we're going to do it on TWiEVO. You want to join us? So here you are.
And so I should actually give a shout out to a gentleman named Pragyesh Dungel. I think I'm pronouncing that right, Dhungel, who's a graduate student in Zilong Yang's lab at Kansas State University, who gave a talk on experiments related to the ones in this paper at ASV. And I approached him afterwards because I was really pumped about this, because this is an exciting topic for me. And he alerted me to the existence of this paper and sent it to me. And so I appreciate his interest in sending me this paper, and I sent it on to you, and here we are, Vincent. Great.
Yeah, very cool. So the paper itself it's titled Trans-Kingdom Mimicry Underlies Ribosome Customization by a Poxvirus Kinase. And the thing that kind of I saw it a few weeks ago. It just came out a few weeks ago in Nature. Senior author is Derek Walsh at Northwestern, the School of Medicine in Chicago. The thing that perked me up was this idea of trans-kingdom mimicry. And in fact, kind of coming at it from an evolutionary genetics perspective, mimicry as an evolutionary strategy kind of pops up all the time. And actually we'll return to that maybe at the very end of our science show today. But then mixing that kind of on the heels of the virology conference, pox viruses, and even I think there's really great energy in ribosome biology continuing on in new ways, which echoes on one of our picks of the week not too long ago from TWiEVO 15. And this was the Harry Noller celebration of his Breakthrough Prize. And a great connection with our guest today, Rich, is a picture from that link in 1976. Do you know the picture, Rich?
I know exactly the picture. I know the picture you're talking about. It's made the rounds because it's such a great picture.
Yeah. Can you give us the backstory?
Sure. So, as I've described before, I got my start in this discipline in Harry Noller's lab as an undergraduate at Santa Cruz. So I graduated from Santa Cruz in 1970. That photo is from 1976, by which time I was a postdoc at the Imperial Cancer Research Fund in Cambridge. And Harry was on sabbatical. And this was actually an amazing sabbatical because he spent part of it in Geneva, where he cloned into Lambda. Okay, this is new. This is 76, so cloning is a very new deal. He cloned into Lambda 16S ribosomal RNA for half of his sabbatical. And then he went to Cambridge to Fred Sanger's lab and learned how to sequence. Okay? And then he went back to Santa Cruz and sequenced 16S ribosomal RNA, which was a big deal. So what a sabbatical at any rate. So he spent some time in Cambridge. That picture, we spent quite a while with Harry while I was there. As a matter of fact, my wife and I took a sort of a 2-week walkabout, or maybe a week, I don't know, in Wales with him. That was just a hoot. During which time those Lambda clones sat in my refrigerator back in Shepherd's Bush in London. At any rate, we visited Harry in Cambridge, and there's a pub in the background, and we had spent the morning in the pub.
Wow.
Or the most of the afternoon, and had that picture taken afterwards. And I think I'm not looking at the picture, but I know it so well from left to right. I believe is my me, and then my wife, and then Harry in his ultra hippie stage, and then a big man with curly hair and a cap, whose name is Bruce Adels, as in Adels sausage.
Yep, right.
In the markets.
Right? Wow.
Yeah.
Wow. Yeah, I know it.
He was a graduate student at Santa Cruz, and was also a postdoc at the ICRF while I was there. And he used to take me out all the time because he was not all that engaged in what he was doing in the laboratory, but loved food. Okay, and he loved to cook, and he would go out. London is just fantastic for this because they got all these little shops, cheese shops and meat shops and wine shops, and Bruce was just crazy about this. So apparently, when he got back to the U.S., he kind of lost interest in the science and figured out he had to make a living for himself somehow. Started making sausage in his kitchen and writing cookbooks, and the rest is history. And I'm sure he could buy and sell all of us many times over.
Yeah, wow.
This picture of you, Rich, I would say this is from your intimidating phase. You have such a mean look on your face.
Well, my mother used to tell me, "You never smile, Richard," and it is a burden. I need to learn to smile. But I had long hair. I had the beard. I've had longer hair than that. So
it's funny.
There you go.
Yeah, when Rich first described this on TWiEVO a long time ago, he said, "Don't share this with anyone." You want anybody to see it, and then it showed up.
Yeah, it's been around. It's a great picture. I love that picture.
It's great.
This paper, by the way, the two first authors are co-first authors, Sujata Jha and Madeline Rollins, are from Northwestern University, and then there's someone from SUNY Albany, and then Peter Sarno's on this paper from Stanford.
Mm-hmm.
Derek Walsh used to be here in New York City, and he used to be with
Ian Moore.
Ian Moore, that's right.
That's right. Yep.
Small world.
Small world.
Right. All right. Let's do this. I have to say, these are beautiful protein gels.
They are.
This is from an era when this is pretty much what we did because there wasn't much else to do. And you could either have crappy protein gels, with crooked lanes and fuzzy bands, or like this really sharp, straight ones.
Well, the amazing thing for me is that I look at this like in figure 1C that we'll get to. I look at the vaccinia infection, and I mean, I recognize it as a vaccinia infection, the protein.
I'm sure.
And I know what a bunch of the proteins are. It's
That's
really
cool.
Really weird.
Yeah.
Now, Nels, as a little background for this, they start out by saying ribosomes have the capacity to selectively control translation, through changes in their composition that enable recognition of specific elements. Can you tell us some examples of that?
Yeah. So, as a sort of non-specialist, but sort of a, very curious person for ribosomal biology, this feels really fresh and new to me. I mean, obviously, I think it'd be great for Rich to weigh in as well. But so that statement got me onto looking at, the citations that they put there. And the first one is a review from Maria Barna's group. So she's at Stanford, and I met her a few years ago at a Pew Scholars meeting, and she gave a really inspiring talk on this idea, in essence, that kind of, and I might be butchering it to some degree, but this idea that ribosomes kind of come in different flavors, or depending on the tissue you're looking at, or the developmental stage of an organism. And by different flavors, what I mean is that this massive protein and RNA complexes coming together in slightly different ways. There are different constituents that may be present or absent or modified in different ways, and that this can have different outcomes in the ways things are translated, or the things that gets translated in the first place. And so in terms of examples, I think there are these, what people are now calling these ribosome opathies, which is when one component might be mutated, that you have very specific outcomes, and in terms of disease phenotypes. And at least I think on the face of it, that's a really big surprise, because, the ribosome is responsible for translating all proteins. And so if that's broken or disabled in some way, you might just on face value predict that you would mess up everything about a cell. When in fact, in the in a metazoan organism, a multicellular organism, there are really specific things that go wrong as you disrupt this. And this, at the very least, hints at this idea that ribosomes might come in many flavors, or that there are different ways that they're modified or deployed in specific ways.
All right. But
I actually have a very personal history with this that goes way back, because my PhD thesis project was supposed to be about bacteriophage T7 tweaking ribosome initiation factors during the course of its infection to change as a part of moderating or mediating changes in gene expression. And it turned out to be hogwash. It was based on a paper. I won't go into the details, but on a published report that said that this was an issue, and a basic part of my thesis was to prove that that was not true. But this came up at a time when initiation factors for protein synthesis and all the other ancillary factors were hot topics and being described in detail, and also comes on the heels of the purification of or the characterization of E. coli RNA polymerase having an initiation factor that could obviously moderate the specificity of the polymerase. And so I think everybody's sights were on tweaking specificity of activity of both polymerase and perhaps ribosomes by messing around with initiation factors or other satellite factors. And I, of course, because of my thesis, really soured on this idea. And it's good to see this actually, freshen up. And this particular paper, in particular, the idea of a virus tweaking the ribosome machinery to its own benefit. Okay. And I don't know if there are any other published examples of that. I've not kept up with this literature.
Yeah, I don't know of any offhand either. I mean, and what I love about that, Rich, is this idea that because of virus, in this case, the pox viruses sort of exploit this or modify this behavior, it helps teach us about how the cell works in the first place, or sort of exposes this layer of ribosome biology that was sort of mysterious, and the viruses sort of show us the way. Yeah, it's really fun.
Right.
Yeah.
So, following the sort of TWiEVO paradigm, my perspective on why this paper is exciting is that we just discussed. It's the idea that a virus will actually tweak the translation machinery in a cell to its own advantage. That is, to preferentially translate the viral messenger RNAs in preference to the cellular messenger RNAs. That's very cool. And it's an idea that's been around forever and happens in different fashions. And also, as we'll get into, there is a long-standing mystery about the structure of a subclass of poxvirus messenger RNAs that's really cool, that this paper solves effectively, or at least addresses in a very positive fashion.
I would say that, and we'll return to this at the end. I would say it's completely accidental. I would guess that this unusual message structure was probably an accident at one point, and it turned out to have this cool advantage.
Yeah,
absolutely.
Yep. Yeah.
Agree. And then that brings us into sort of the TWiEVO part of it too on the backside, which is this idea that will come up of mimicry and how that connects through the biology. And then I think we'll end up in we'll see where the conversation takes us, but I think we'll end up in a really fun sort of hybrid scenario, one of our TWiEVO episodes here, where we cross over between the two.
Is it? TWiEV infects TWiEVO?
There you go. I like it. So I propose that we kind of go through what starts out, I would say, as really a virology paper with cool implications for ribosomal biology, and then return to the evolutionary sort of view.
So Nels, tell us, I think the way to start is to tell us about RAC1.
Yeah, and so this kind of comes out of left field, at least it did for me as I was reading the paper, is that, so basically, as we're talking about different flavors of ribosomes or different kind of conglomerations of factors coming together, they draw a connection to a small ribosomal protein that is known to have some influence on translation called RAC1. And so I had to do a little digging. How did they get on to RAC1 versus all of the other hundreds of components that come together? And so it turns out that RAC1 was actually identified for a connection to viruses from a genetic screen done in Drosophila. This was done by Sarah Cherry when she was a postdoc in the Norbert Perelman's lab. And actually, you mentioned that science is a small world. Sarah, we just saw her at ASV. She was in that session.
Yep, that's right.
On virus evolution. Another little fun connection there. But so what they did is, or what Sarah did and continues to do, and she spoke about recently in Madison, is to knock down many genes individually in Drosophila cells, and then test whether that has an impact on the ability of a virus to replicate in them. And I was looking back at that reference. They actually uncovered, I think, more than 60 ribosome-associated or components or associated proteins, these auxiliary proteins in some cases, that they tabbed as being important for these iris sites, or these internal ribosome entry sites that some classes of RNA viruses in particular have, that allow for cap-independent translation.
Mm-hmm. We used to work on irises because our picornaviruses all have them. Internal ribosome entry. You don't need a cap. You don't need a 5' end either. So RAC1 is needed for iris-mediated translation, correct?
Correct. Yep. And so then, but these are very different classes of viruses. The RNA viruses that use these internal entry sites in order to sort of fool, if you want, the ribosome into translating viral transcripts. And so the question the authors came at with this is whether there might be a connection to DNA virus protein translation as well. And already this is very different, because these are capped transcripts. And maybe we should just take a minute to just define what we're talking about here between capped and uncapped transcripts. And I couldn't imagine a better person than Rich Condit to jump in on that.
Oh, my goodness. Okay. So see if I can do this. So the cap on an RNA is a guanine residue that is put onto the 5' end, which is, I don't know, the left end. It's the end where translation starts. Okay, the cap is a special nucleotide. Well, it's a guanosine triphosphate that is stuck onto the 5' end chemically backwards. Okay, so it's a 5' linkage. So for the uninitiated, just think of it, and it's a triphosphate linkage rather than a 3' 5' monophosphate. It's a 5' triphosphate. So for the uninitiated, just think of it as a pretty silly nucleotide stuck dangling on the front end of a messenger RNA, and it's further modified because it's methylated. Okay, and there's a whole apparatus that does this, and ribosomes. There are factors associated with ribosomes. The cap does two things. It enhances translation of the messenger RNA, and it also confers stability as a sort of a blocking group on the 5' end. And in terms of enhancing translation, the dogma is that the way translation happens in eukaryotes, by the way, caps don't exist in prokaryotes, and translation initiation happens by a somewhat different mechanism. But in eukaryotes, there are specialized initiation factors for translation that actually recognize that silly capped nucleotide and bind to it, and that's how the ribosomes grab onto the messenger RNA. And then there is what is called, and I think this is relevant to this paper as well, a 5' untranslated region. Okay, so there's a sequence of nucleotides that does not code for protein that the ribosome must get through before it gets to the initiating nucleotide, which is always, well, almost always an AUG encoding methionine. And the 5' untranslated region can be as short as maybe 30 nucleotides and as long as hundreds of nucleotides. And the thinking is that the ribosome binds to the cap and then does what's called scanning, which is to slide down this 5' untranslated region until it encounters the first AUG that is in a favorable context, okay? That is, it's got flanking nucleotides that base pair with a ribosomal RNA and that kind of stuff, and it says, aha, this is where I start. And I think it's also important to note that at that point, where the ribosome is starting translation, it's got the AUG there, and that's kind of in the middle of this giant macromolecular machine. Okay, it's got like 50 proteins and two big subunits that have 3 or 4 different RNA molecules. It's a giant macromolecular machine. So that's binding the RNA at the AUG, and the footprint, that is the amount of RNA that's actually bound, is about, I think, 30 or 40 nucleotides, with the AUG sitting just in the middle. So there is some upstream untranslated sequence actually bound by the ribosome. All right, this is going to become important later on. In an uncapped message, and we're talking, for example, in poliovirus and several other positive strand viruses, you may have on the messenger RNA a protein on the 5' end rather than a cap. And there is in the 5' untranslated region, there is a specialized structure that's got a bunch of base pairing and stuff called the internal ribosome entry site that serves to attract and bind ribosomes in a cap-independent fashion. The ribosomes can bind that sequence without having to see a cap, and then do the same thing, scan to the AUG and start. Did I get all that right?
It's perfect.
Yeah.
And RAC1 is a protein that helps this internal ribosome entry happen in some way. And I want to add something interesting. Some plant viruses that are not capped, but that have a protein at the 5' end of the RNA, they're plant picorna-like viruses. The protein actually recruits the ribosome There's no iris. There's actually a protein recruited right to the 5' end. So there are many ways to solve the capless problem.
Yeah, it's cool.
So I was puzzled. My first reaction to this RAC1, because it kind of in the paper, they've got a sort of general introduction into translation or regulating translation, and then all of a sudden RAC1 just pops out. It'd be interesting to hear the backstory on this and how they actually got to this. My guess is they had some indication.
Yeah, it must have.
That RAC1 was involved in regulation of translation and said, "Ooh, let's knock this out and see what happens."
Yeah, because they say to determine if RAC1, is involved in translation of DNA viral mRNAs. It's like, why would you do that?
Right.
That's right. Yep.
So the first thing that startled me was that you could actually knock this thing out and still get translation to happen.
That's
right. So that got me into looking into RAC1, and it is in fact an integral ribosomal protein. That is, this is not some peripheral factor or anything like that. It's highly conserved and associated with all ribosomes. It sits on the surface of, was it the large or the small subunit, I think, near the RNA Exit Channel. Okay, but you can knock it out and translation still happens. But as is said here, that ribosome without RAC1 won't translate an RNA with an iris element, but will translate other RNAs, right?
Yep.
Okay.
And then the authors kind of present us with maybe a second mystery, but one that's more kind of that we can sink our teeth into. Is in order to test this idea, does it matter for DNA virus translation? What they did is they took a haploid cell line, the so-called HAP1 line, with a knockout of the RAC1 gene, and then asked, does this influence translation for two different very distinct DNA viruses? So vaccinia virus, the model pox virus, which both Rich and I are big fans of, and then secondly, herpes viruses. And so what these two viruses have in common is that they both encode or deploy cap transcripts. But the result, which is pretty striking, and that gets back to those gorgeous protein gels that you both mentioned a little bit back, is that vaccinia translation is down, but herpes virus translation is okay.
But it's late translation, right?
Indeed.
Not early. So the early, and maybe Rich can tell us a little bit about the temporal. Early vaccinias are fine, right?
Right. So, they show in a later part of the figure that early gene expression is okay, and late gene expression is not. Actually, in a vaccinia infection, there are three distinct classes of transcription. Early transcript. So this virus is unusual because it replicates from a DNA genome in the cytoplasm of infected cells. That means that it has to carry into the cytoplasm in the virus particle all of the machinery necessary to do early transcription. In fact, in the case of vaccinia, all that machinery is also virus coded. In the previous round of infection, you make polymerase, capping enzyme, poly-A polymerase, all of this stuff, put it in the particle to deliver that whole package to the next infection, and that uncoats in the cytoplasm, and in this subvirion particle, you get early transcription. And then some of the proteins that are made early during infection modify the specificity of the virus RNA polymerase so that it now recognizes a new class of promoters, which are intermediate promoters. And some of the gene products from intermediate promoters are yet another class of bunch of transacting factors that once again modify the RNA polymerase to now recognize a third class of promoters called late promoters. And this gives you a scheduled sequence of gene expression from early, intermediate, and late, and appropriate proteins are made to time the infection right at each of those stages. Importantly for this, they don't make the distinction in this paper between intermediate and late. They just talk about late. I used to use the word post-replicative to include both intermediate and late. But for the purposes of this paper, intermediate and late transcription is similar enough so that they can be lumped together and call them late. In particular, the unusual structure of the messages that we'll get to in the intermediate and late RNAs are the same.
Got it. All right. The next experiment is they use RNAi to deplete RAC1 in primary cells. Now, this is different. You're not knocking out the gene. You're just knocking down the amount of RNA. And surprisingly, there's no effect on vaccinia translation.
Yeah, and I like this a lot because this is sort of an important clue that got them, allowed them to figure out what was going on with vaccinia, or at least sort of get them on to the next insight. And part of this, I mean, it's kind of maybe a peripheral side note, but this idea that, if you disrupt a gene in different ways, there can be different outcomes or different phenotypes. And so an RNA,
yes.
Yeah, exact. So an RNAi knockdown, where there's still a little bit of, in this case, the RAC1 transcript to make RAC1 protein, ended up being the complete opposite result from the complete disruption of the gene or the knockout of it in the other cell line. And so the reason this was important was that they actually, when they looked at RAC1, the protein on a gel, and this is now the bottom of panel of figure 1C, they noticed that RAC1 wasn't just migrating as a single protein. There was actually this doublet, and they highlight this. And in the knockdown cells in particular, the doublet was now just turning back into a singlet, but one that was migrating more slowly through the gel than the lower band. And what this implies is that there is a modification to the RAC1 protein that might be really important for the ability of vaccinia to translate its proteins.
Mm-hmm.
Right. So in the mock-infected cells, there's a doublet that's the slower and the faster migrating guy, and in the vaccinia-infected cells in the knockdown, there is only the slower guy. So maybe that slower guy is what allows vaccinia to get through the infection.
Mm-hmm.
I've seen this bit with knockdowns not working like knockouts frequently.
Mm.
Because you're not knocking it all the way out. You're just knocking it down.
Yep.
And in many cases, a little bit is enough.
Yeah, and I think in some cases, in sort of the emerging CRISPR era, we sometimes gloss over this idea that, we'll just knock everything out and we'll understand all the biology. Whereas here is a perfect example of how knocking something down gave this really cool clue to how this protein might be modified. Yeah.
Mm-hmm.
The next step is something that, 20, 15 years ago would have taken your lifetime to do. But now with liquid chromatography and mass spectrometry pretty much available to every lab, you could take a protein and say, what's unusual about it? So they did that, and they found there's a series of 4 amino acids, serines and threonines. Serine, threonine, serine, threonine, right in a row, that are phosphorylated in vaccinia-infected cells. And, one phosphorylation per peptide with the position 278, which is a serine, is the one that's most likely phosphorylated. So single site modification in vaccinia virus infected cell, and that probably explains the doublet, right?
Right.
Mm-hmm, mm-hmm, mm-hmm.
All right, so that's a change, and that's a big deal. It takes a long time to do that.
So I misspoke back there because I said the doublet was in mock-infected cells. No, the doublet is specific to vaccinia-infected cells.
That's right, because vaccinia is phosphorylating somehow in vaccinia-infected cells. We don't know how it's happening. This protein is being phosphorylated on a string of serines and threonines, and specifically at one residue, serine 278 most of the time, single-site modification.
God, I love the way the logic in this progresses. This is great.
And then they have a binding protein, which is called PKC beta 2, which is that protein kinase C, I guess, right?
Yeah, I think that's correct. One of the sort of isoforms or different flavors of that kinase, the host kinase. Yeah,
that binds RAC1. And if you inhibit PKC beta 2, it inhibits phosphorylation of RAC1. And that, of course, isn't good for vaccinia translation when you do that. Mm-hmm. But they said this allowed them to come to the conclusion that if you don't have any RAC1, you inhibit vaccinia translation, vaccinia mRNA translation. But if you have a little bit, it can be modified, and you don't need it all modified, just enough to get translation going.
Right.
And I think this PKC beta 2 thing was a key part of that, right? Because they could show that if you inhibited its function, and even if you had RAC2 around, you had to have a little bit of phosphorylation in order for translation to occur.
Yeah, that's right. And then this ties back to the point that Rich just made, that this is, where they see that that slowly migrating version or modified version of RAC1 is in the context of a vaccinia infection. And so there are a couple of kinases encoded by the virus itself, which then become sort of prime, candidates for being the virus's part of this modification of RAC1. So there's, I think the B1 is one of them, and the F10 are two proteins that are kinases that are virus encoded. And so they move on to test whether in mutants, viruses that are mutants of either the B1 gene encoding the B1 protein or F10, whether that influences the migration of RAC1 on a gel. And so this is now, we're into the second figure, panel 2A. And what they can see is that the F10 mutants, that doublet has moved to a singlet that's that modified version that they saw in the knockdown experiment. However, the B1 mutant is now a singlet that does not migrate more slowly. It's not modified. And so that makes a strong connection between B1 as being the virus-encoded activity that's modifying or phosphorylating the RAC1 protein.
Rich, only two kinases in the genome, is that right?
I believe that's correct. B1 and F10. And B1 has, both of them are very interesting proteins. B1 has a very interesting history and phenotype. It's been worked on by the Trackman lab a lot. But the phenotype of the B1 mutants is that they can't replicate DNA. Mm-hmm. And it's because when vaccinia DNA goes into the cell, it's bound by a cellular protein called BAF that inhibits its replication. And the B1 kinase phosphorylates BAF and inactivates its DNA binding activity so that the vaccinia DNA is available for replication. Really weird.
I want to point out...
B1 does a bunch of... Both of these kinases phosphorylate lots of stuff.
Yeah, multitaskers.
I want to point out one observation before we go on.
Yep.
So if you inhibit phosphorylation of RAC1 by using a PKC beta 2 inhibitor, you don't get host. Not only do you not get vaccinia mRNA translation, but you do not get host translational shutoff, which I understand occurs late in pox virus infection, correct?
Host translational shutoff?
Yep.
It goes in phases throughout the infection, really. But it's you're right that it's most dramatic late during infection, and that's due to a bunch of stuff, not the least of which is the virus makes decapping enzymes. So this is going to make it really complicated, that decap a bunch of host messenger RNAs and catalyze their degradation.
So this is important. It'll come back later. Again, if you don't phosphorylate RAC1, not only do you not get vaccinia mRNA translation, but you don't get shut off. You don't get shut off.
Yes, right.
Very important.
Right.
So B1 is what they think is phosphorylating RAC1, right, Nels?
Yeah, exactly. On the virus side of this. And then, getting back to Rich's point that it's a multitasker, that it's actually has all of these targets. They actually, in order to, do the experiment, they had to use what's known sort of in the field as a temperature sensitive mutation of B1, just so that you can get virus to replicate at all. But in fact, it turns out that this connects back to the host biology, that because you have that protein kinase C isoform involved, that's really what's going on here. And so it's not that it's a temperature sensitive mutation. It's just there's sort of more functions in the background that allow some of the process to move forward, and sort of almost this molecular conversation between the virus and the host proteins modifying RAC1 as part of the biology here. Yeah.
So, they said in the paper that temperature sensitive is a misnomer, and I have to say, I bristled a little bit at that. I want to just to beat my chest here a little bit. All these mutants are mutants that I made.
That's right.
CTS2 I probably made in Stony Brook in 1978. So
Wow.
I numbered them in sequence when I made them. Okay.
Cool.
So that was the second TS mutant that I made. At any rate, yeah, I mean, they're temperature sensitive. They form plaques at a low temperature, not at a high temperature. That's temperature sensitive.
Yep.
Okay. But when you do the analysis further, and it's the Trackman lab that did most of this, you find out that the B1 protein is unstable. A common mechanism of temperature sensitivity is that you raise the temperature on a mutant protein and it gets degraded. But in this particular case, the mutation causes the protein to be degraded at both a low temperature and a high temperature. And so the temperature sensitivity must be more complicated than that. The protein is absent under both circumstances, and so the temperature sensitivity must have to do with, say, there's some sort of host kinase that can substitute for B1 at a low temperature, but not at a high temperature, or something like that.
Yeah. Yep. Yeah, and that connects back to that protein kinase C beta 2.
Right.
They also made a fluorescent protein fusion of B1 and show that it co-localizes probably with RAC1 in these viral assembly factories, which is also good to know, besides it being a kinase, right? It's in the right place.
Did you look at all these movies?
They're very nice.
Yeah. So the factories are interesting. These are the factories are that's where the viral DNA replicates in the cytoplasm, and they start off as small. If you look at DAPI staining, which stains DNA, to examine these things, they start out small and very regular in shape, round, and very uniform in appearance according to the DNA stain. Those are in the early stages of DNA replication, and then they grow, and towards the late stages, they take on a much more sort of pleomorphic, that is irregular shape. And if you look at the DAPI staining, it has holes in it, dark spots. Okay. And if you stain for other things like messenger RNA or ribosome initiation factors or newly synthesized viral proteins, you find they're filling those holes. So there's a whole bunch of DNA with cavities in it, and there's a whole bunch of activity going on in those cavities, most likely translation. And there's more translation that goes on in the periphery of these factories as well.
Rich, they mentioned that so if you inhibit phosphorylation of RAC, it blocks formation of these factories. So why would that be if the translational block is not early, but it's middle to late, right?
Okay, so
are these assembled later in infection?
No, actually, that I'm puzzled by that, because they say they don't make the factories. They don't make the late factories. They don't even make early factories. No, they make the early factories, right?
It says prevented the formation of factories.
Of factories.
That's all they say.
Okay.
Yeah.
Okay. So that implies that you're not even getting any DNA replication. Okay. And so, but you are getting early protein synthesis. If you're getting early protein synthesis, I would anticipate that you would get DNA replication and you would get the formation of factories. I mean, you see that all the time. So there must be I can't explain that unless there's some effect on some early protein, or maybe there's some trick with an intermediate protein being required for the full progression through DNA replication. That would surprise. That doesn't make any sense to me. Because that flies in the face of other pox virology. So I'll have to think about it, but I can't explain that one.
Okay. Yeah. All right. Not essential for our story here.
Right.
True. Yep.
Okay.
Yeah. So then, so I'd say up till now, they've, what at least for me is a pretty convincing argument based on the localization, maybe even more convincingly based on directly based on the difference in RAC1 migration in those virus mutant backgrounds, the B1 and F10. Then the question kind of shifts to the other part of this biology, which are the transcripts, the viral transcripts themselves, and how they might be recognized by this modified version or phosphorylated version of RAC1. And so this brings us to then looking, or for the authors looking specifically at the transcripts themselves, and this fact that they have these basically poly A, 5' poly A regions in the cap near the capped region of the transcript. So this is pretty strange, right? So to have a run of adenosines in a transcript and specific to the virus transcripts, and so they actually are able to test whether these poly A regions matter for the translation of those transcripts by using a luciferase reporter activity. So basically, the way this experiment is set up is you take a gene encoding luciferase, this enzyme that has very clear activity that you can measure by fluorescence markers, and then they hook it up to poly A regions, 5' poly A regions, or non-poly A regions, and then ask whether it matters if RAC1 is modified or not, or in different flavors. And so maybe some of the early, most definitive evidence that they show is that in a mock situation where you just have a beta-actin promoter or 5' region, there's no difference in the luciferase reporter. If you put a poly U string, so instead of a poly A string, there's also no increase. So this is in either mock or vaccinia virus infected cells. However, if they put a reporter transcript that has a poly A 5' region, and then also infect those, now all of a sudden it spikes up the amount of the reporter, the luciferase reporter that you see. This is in figure 3D. And so this connects basically the two phenotypes, or the RAC1 modification to the viral transcripts in their translation.
So, in the paper, they go right to this poly A leader. I just wonder how they reached it,
Well, these guys are not unfamiliar with this system. Ian Moore's worked on and published on vaccinia gene expression in the past. So they're probably quite aware of this. And as a matter of fact, they had to be thinking this from the very beginning when they were comparing herpes and
I suppose
infection.
That's the main. That's the main difference, right?
Right. So I want to talk about this poly A leader, which is sometimes called a poly A head as well, because this is the thing that has been around for a while in vaccinia. That's a big mystery, or has been a big mystery. Mm-hmm. So let's go back to transcription. Ordinarily, remember I said that in messenger RNAs, there's a 5' untranslated region. And that results from the fact that when RNA polymerase initiates transcription on a DNA template, it initiates transcription usually on the order of 30 nucleotides and maybe as little as maybe 15 or 20, but usually on the order of 30 nucleotides, or it could be even hundreds of nucleotides upstream from where translation is going to actually start. And it synthesizes a transcript from the template, that has this 5' untranslated region that gets capped, and then you get to the AUG. Well, for intermediate and late promoters in vaccinia, that's not what happens. It happens differently. The way you recognize an intermediate or a late promoter in vaccinia is look for upstream from the AUG that is probably the first codon, the initiating AUG. Upstream from that, you look for a TAAA sequence, either T and 3 A's or T and 4 A's. Okay, and that is a dead giveaway that that's going to be an intermediate or a late promoter. And then the whole composition of the promoters, there's a spacer region upstream from that, and then upstream from that, there's an AT rich region. So the whole promoter is a sequence of about 20 nucleotides that the polymerase recognizes. But the initiating nucleotide is kind of, sort of, in that AAA region, which on the template strand is a triple T. Okay, so what happens is that the polymerase initiates transcription, say, at the upstream most of those guys, and synthesizes, say, the 3A residues, the last of which would be the first A in the AUG, okay? And then, without letting go of that transcript, slips backwards and then retranscribes that, tacking it on to the initial 3As, and does that multiple times to generate this effectively non-templated poly-A head. That poly-A is not encoded as a string of A's in the DNA. It's just this 3 or 4 A residues right upstream from the AUG, and the polymerase is doing what I call stuttering, going back and forth and spitting out this poly-A head that eventually gets capped. And this to me is brilliant. Because, I mean, why waste all that DNA making a 5' non-coded region, right? When you can just, you got to have, I presume you got to have something. You can't just cap the AUG. That's not going to work. You got to have a 5' untranslated region for the ribosome to grab onto. So in this slippage mechanism on this stretch of T residues on the template strand, stutters back and forth and spews out this non-templated poly-A head that gets capped. And that's a very, very unusual thing. I don't know of any other situation where that happens other than in poxvirus.
So it's the 5' UTR is entirely A?
Yeah.
There's nothing else?
No. It's all A's.
And
it's heterogeneous in size.
Uh-huh.
I think the average size is about 15 nucleotides, but it can be up to 30 or 40.
You know, you can't argue that it's saving room in the genome because the bloody genome is huge, right?
Yeah, right. I always thought of it that way, but I agree that and clearly from this, in terms of evolutionary value, there's other things going on. But this poly-A head, which was described initially in the early 80s, I think, late 70s or early 80s by two groups, Henk Stunnenberg and Rico Wittek, has for 30 years been a mystery. Why is this there? Okay, because it's weird.
Now, again, you may have said this. Is this just late RNAs or all mRNAs?
Okay, so I'm glad you asked that again. It's intermediate and late RNAs.
Okay, got it.
But going back to the previous question that I couldn't answer, they don't ask in these experiments whether in the RAC1 knockout the virus can actually make DNA, whether it's DNA negative. And they only check for a couple of early proteins. So I would want to look in much more, in order to understand why you don't get factories, I'd want to look in much more detail at early protein synthesis and at DNA replication. Because if I'm not mistaken, there are a couple of early genes that have poly-A leaders on them.
Oh, interesting. Yeah, those would be the perfect ones.
I'm not absolutely certain about that, but if that's the case, it could be that you don't make the complete set of early proteins, and if some of those guys that you don't make are important for DNA replication, that would explain why you don't make factories.
Yep.
Okay.
Good point.
Cool. Yeah. Yep.
I hope they're listening.
That's right. Yeah, check that out.
I hope we get some. I hope we get some mail here. I should know the answers to all these questions, but I don't.
No, that's great. So, and like you say, Rich, I mean, so now like a really cool 30-year mystery basically solved, I think.
Absolutely.
And so you could imagine that would be enough that you could, kind of pat yourself on the back, maybe send this in, get it published. But then the authors go on, and here's where I would say the sort of TWiEVO hybrid scenario comes in, to then take a look at, so to go back to that RAC1 protein that's involved in recognizing these poly-A leaders, and ask what's going on in a phylogenetic sense. That is to compare RAC1, this highly conserved protein, among lots of different species, and see if there are any clues there in that comparison that might uncover what's going on here with this interaction with the virus, or how this biology might be working, this recognition of these modified transcripts by the ribosome. And so if they have, so luckily there's first of all some structural data for this known, so they can map these serine and threonine sites, the so-called SSTS loop, STSS loop, and see where that sits, and then make a robust alignment with many other species. And so they have the human sequence, yeast sequence, looks like the Arabidopsis in tobacco for plants, and then they also have C. elegans, mouse, and Drosophila. And so what they see is basically from human to worms or flies that STSS loop is quite well conserved. Most of the serines and threonines are all there and can be aligned. But then what you see in plants is sort of strikingly different. There's actually an insertion into that loop of more amino acid residues. And they're not just sort of random residues. They note that there are some glutamic acid, some negatively charged residues. And when you put that together with the fact that the serines and threonines, these are the residues that are getting phosphorylated by the virus kinase, that act of phosphorylation puts a negative charge in those locations. And so this is where this idea of trans-kingdom mimicry that they put in the kind of provocatively in the title of the paper is that basically the plant version of RAC1 already has these negative residues there as if it's already been phosphorylated.
The authors are calling you.
That's right. That's right. I had one of my buddies call for sort of dramatic for dramatic emphasis of this evolutionary comparison. Sorry for that interruption. I think we're okay. And so this allows them to sort of ask another kind of interesting question along these lines, which is, okay, well, what happens if we go back to the human version and basically replace it or make a chimeric molecule where they actually insert that loop from the plants, or take the serine, threonine, serines, and then do a substitution to a glutamic acid, which is often considered by biochemists to be a phosphomimetic substitution, meaning that instead of phosphorylating, if you just change to that residue, that you'll, in essence, the R group will approximate in that amino acid will approximate a phosphorylation event. And then what they can do is to, go back to these reporters, those luciferase reporters with the 5' leaders, and then see what that does to the translation of those proteins as judged by the luciferase activity. And so what they observe is actually a pretty clear enhancement of translation if they take the entire plant protein, or just substitute in that small insertion in that one loop of the protein, or just make a single substitution to a glutamic acid at one of the serines of the 3 in that loop. In all 3 cases, they actually see that poly A transcript is translated much more efficiently.
What I think is cool is that you don't want to put a phosphomimetic throughout the whole sequence. It's better if you just put one in.
Yeah.
Right? Single site phosphomimetic.
Right.
And they say,
right,
as single sites, are phosphorylated, this highlights the exquisite refinement with which the viral kinase modifies RAC to mimic the plant counterpart. I just thought that was so cool. Right.
So, can either of you guys tell me what's unusual about plant messenger RNAs?
Exactly. I don't know. Are there poly A containing plant? I don't know.
Great question. I had this same question. Why is it there? And in fact, I would have. I think it's great that they kind of went to this extra step to do this simple evolutionary comparison, because it got them to that observation of this sort of refined activity, or the idea that you can just modify a single site. They could have actually, I think, pushed this a little bit farther. So, they compared out to plants. If you add other eukaryote versions of RAC1, there could be some additional information in there. So, for example, is it that plants really are doing something strange, maybe with a poly A or another modification to their transcripts? Or do you see that in all of the diversity of single cell eukaryotes out there? Maybe there are some more clues about sort of, who's mimicking who in a sense, or is there other biology happening here? And so that's where I thought, I love the idea, and I love that it got them to this sort of hypothesis that the kinase has to just make these very single modifications. And if it modifies all 4, you actually see a reduction in some of the enhancement or other consequences. But in terms of actually nailing down who's mimicking who, or if this is really mimicry. I think that still remains to be explored, which is also an interesting future direction.
I think it would be neat to take out these charged amino acids from plants and see if there's a problem.
Mm-hmm.
Right?
Agree.
Yeah.
Yep.
I mean, maybe just a few, maybe a subset of messages would be affected. You could see which ones they are, maybe, because they have a way of knowing which messages are associated with RAC1.
Mm-hmm.
In fact, that's their assay. So you could say, take out the charged sequence in the plant RAC1 and see if certain messages are not translated. Anyway.
Agree. Yeah. Cool idea. Yep.
Sorry, Rich, you were going to say something.
I was just going to ask Nels, when you hear the word mimicry, what happens in your brain? Okay,
he gets all excited
because it sounds very deliberate. You know.
Yeah.
Okay.
It's a good point.
So how do you think of the word? How do you think of the word mimicry in terms of evolution?
Yeah. So certainly, it's a great question. I mean, classically, mimicry, I would say, was introduced as an idea, back in the Brazilian rainforest in the 1850s with these observations of Mullerian and Batesian mimicry, where these butterflies approximate each other's appearance so basically edible butterflies mimicking the appearance of a poisonous one to fool predators into not eating them as a snack. And so the idea of mimicry in evolution has, first of all, a really long history. There are all kinds of, very sort of almost formalized definitions of it, and how some of the the original observations in ecological settings, like with butterflies mimicking the appearance, applies to these sort of molecular, down to a 4 amino acid loop that is negatively charged. I would exercise some caution in kind of defining this as mimicry before you really know, a little bit of that phylogenetic context. Where did this come up? What is this Vincent's experiment? What does this mean for plants and how they're translating things, to really know whether there's a functional mimicry that's happening here, or if it's sort of a coincidence in a sense of, so the biology kind of converging, and that these are important ways of regulating protein translation. And so, I would say, at least my take on this would be that it's a provocative and interesting title. Have they really nailed the idea that this is mimicry? I think we would have to know more about the biology behind it.
So when you give the butterfly example, it's obvious to me how natural selection would work to move that mimicry forward. Okay,
Mm-hmm.
if you look like the poisonous butterfly, you've got a better survival advantage. It's not obvious to me how there would be some sort of selection at work to result in what's being called mimicry here. Other than the only thing I can think of is that it's fairly clear from this that messing with the charge content on this loop in RAC1 influences how messenger RNAs can be translated, in particular with respect to what's in the upstream untranslated region. Okay, and so you can see how that, as a general mechanism, might play out to modifying that loop in different systems to give preferential translation of one or another transcript.
Yeah, I agree, Rich. And I mean, for example, one experiment that might start to, go in this direction would actually be to test whether, in the case where you make the phosphomimetic substitutions or substitute out or in the plant sequence, does that actually have an impact on vaccinia virus replication? If you infect those modified cells encoding those modified proteins, does that actually influence virus fitness in any way, which they haven't tested yet? I would say that, the fact that you have this like very clear kind of an interesting connection between what's happening in that very same loop in plants does make a nice case that basically lightning has struck at least twice here. That,
right.
And then that implies that there is something going on. What that is and whether this is truly mimicry, I think remains to be seen. Are there plant viruses that have sort of tapped into RAC1 in plants, and then that caused that very different outcome in terms of the insertion and deletion. Has in mammalian systems did there used to be something that looked like the plant version, then that was lost as a counter adaptation potentially to some ancient pox virus like ancestral virus, or is there just sort of more evolutionary noise and there isn't so much of an adaptive scenario in either or both cases? I think that remains to be seen. Yeah.
Rich, is there any insight into vaccinia-mediated host shutoff of translation from this? Because we said earlier that blocking RAC phosphorylation inhibits host shutoff. So do you see any connection there?
Well, if going back to our previous conversation, if DNA replication is inhibited. So as I said, shutoff kind of goes in phases. There's a little bit of shutoff that happens early, and then when you get to the late phase, everything crashes. And that's partly because these decapping enzymes are intermediate and late gene products. Okay. Mm-hmm. So if in fact, and post-replicative gene expression is coupled to DNA replication, and obviously from this, translation of intermediate and late gene products is RAC1 dependent. So in this particular infection, if in fact DNA replication is not even happening, that's going to mean that the vaccinia proteins that are responsible for shutoff late during infection are getting made.
Okay. So it's an indirect effect.
Yeah, Got it. Indirect effect.
The other question is what's happening between the phosphorylated RAC and the poly A?
Well, that's, they say that, well, I don't quite, that RAC1 sits at the RNA exit channel.
Yeah.
Okay.
Kind of late there.
So I don't know enough, I don't know ' ribosome biology to know what that means. But the RNA exit channel must be where the 5' end of the RNA emerges from the ribosome, right? Mm-hmm. So when you're talking about initiating, that's the right place to be. Okay, so I've been assuming all along, geez, I should have pinged Harry. I've been assuming all along that loop on RAC1 interacts with the 5' untranslated region. Okay, now how a negative charge would influence that, and how it would specifically influence it in a fashion based on so how it would specifically recognize or a poly A region, I don't know. Okay, but my assumption would be that if you did the appropriate experiments, and I'll bet you, Harry's done all kinds of cross-linking experiments and that kind of stuff. I'll bet you that loop cross-links with the 5' untranslated region in an initiation complex. Geez, if that were the case, they would have said that in this paper.
Yeah.
So there's an experiment.
Agree. And there might be another way maybe of getting at this too, Vincent, is there might actually be a clue from some of the plant viruses. So the authors do go on right in the kind of at the end of the paper to point out that tobacco mosaic virus has what is known as an omega leader, 5' leader, which consists of CAA repeats.
Right.
And then they also note that these leaders exhibit little or no enhancer activity in mammalian systems, but function efficiently in plants. And so perhaps there's a clue there about how the CAA repeats interact with that, those negatively charged insertion part of that loop, compared to how the poly A leaders in vaccinia are interacting.
But that's tobacco mosaic virus. It's not plant cell mRNAs, right?
Right.
Because
good point.
I don't think it's there for the virus, right?
No, right, exactly.
So there must be some host advantage to having that extra negatively charged RAC1 loop.
When we're done with this, I'm going to email this paper to Harry.
Cool.
And ask him about RAC1, because I'll bet you, he's got all the models sitting right in front of him, and he'll know the answer right off the top of his head. And I'll get back to you.
Rich, we should get Harry on one of our shows.
Oh, we should. He's he's such a hoot. He's a good time. So one other just really amusing minor thing. They call in the paper, they call this 5' poly A head, they call its generation through this stuttering mechanism, they call it erroneous transcription.
Mm-hmm.
I bristle at that as well, okay? Because it's interesting to think, yeah, I mean, we were talking in the pre-show about how initially that was probably an accident, okay? But, just like in science, that sort of serendipity can lead to good things. And I'm sure that it had survival value. Okay. So in the long run, in an evolutionary scale, that's not erroneous. Okay. That's the way the virus does it. And it's anthropomorphically deliberate, right?
Well, at one time, a long time ago,
it was a boo-boo.
It may have been a boo-boo that turned out to have selective advantage, right?
Exactly. But that's the way evolution works, right?
It's not erroneous. Exactly. It's not erroneous to get efficiently translated mRNAs.
Yeah. So I just want to make sure that, Nels can do the evolutionary thing. I want to make sure in the summary here that the virology gets across, because it may get lost in the details. So the virus makes a specialized kind of RNA late during infection that has these 5' poly A heads, and it ensures that those will be preferentially translated relative to host messenger RNAs late during infection by using a viral kinase to phosphorylate this loop on the RAC1 protein in the ribosomes, so that now the ribosomes preferentially recognize that specialized vaccinia messenger RNA relative to host RNA, and that sort of preference in translation would give the virus an advantage in the cells.
Is that itself a form of host shutoff?
I would think so, yeah.
So maybe if you take away the advantage, then you lose. Does that make sense?
Okay. Yes, that makes sense. That in itself is a form of host shutoff. Yes, should have thought of that earlier, but that's right.
Yeah, by gaining preferential access to the ribosome, the host.
Right.
Yep.
Right.
And that's why when you block phosphorylation of RAC1, there's no more host shutoff, because everybody's translated at the same efficiency then, right?
The host shutoff thing is complicated because there's it's a multi-part thing. There are many things going on, but you're absolutely right. A suppression of host translation, that is host shutoff, would be a direct consequence, according to these data, would be a direct consequence of the phosphorylation of RAC1. That's right.
Yeah. So, and then I would pick up, I guess, from an evolutionary standpoint and say that, even just doing that simple, going kind of a little bit farther by doing that simple comparison with RAC1 variation among different eukaryotes, or from humans to plants, gave this really nice insight into how in these other cases that you have already these kind of negatively charged amino acids that can kind of approximate that same activity. And in this case, I think specifically what it helped them illuminate was this idea from that very first observation that when they did the mass spec, it was just a single residue that was getting phosphorylated. And that as they played around with the negative charges or the phosphorylation of the serines and threonines, that in fact, it looks like that kinase is somehow just doing single phosphorylation events, even though they're targets for up to 4 sites right in that those 4 residues in a row in that loop. And so it was kind of by making that comparison to the plant version that insight came out that this could actually be sort of have been refined on, and then they go on to say that, through precise phosphorylation events, poxvirus kinase converts human ribosomes to a plant-like state. And so I like that, and it's provocative. It kind of gets at this idea of trans-kingdom mimicry, but I think we already discussed a little bit how it would be nice to, flesh that out a bit more. But what an interesting paper, I think, all around.
Very cool. Very, very cool.
You know what? Something just leapt to mind, and this blows my mind. There was, talking about host shutoff, there was a guy named Rostam Bablanian, a poxvirologist from way back, who was was trying to understand shutoff of host protein synthesis in vaccinia infected cells. And through a series of experiments that start with, if you heavily UV irradiate vaccinia and infect cells with it, you still get host shutoff. But the only thing the virus makes is these a bunch of short poly-A sequences. Mm-hmm. And I think that was the observation. At any rate, Babylonian came up with the idea that these things that he called pollads, okay, with these short poly-A sequences were responsible for shutting off host protein synthesis. He has a bunch of in vitro translation experiments to prove this. And
might be right.
I'm sorry, Ross, but we all thought he was nuts. Because, come on, these polyRNA, this was before anybody knew about polyads or anything like that. And, but I'll bet you he was onto something. Huh. I mean, certainly in the in vitro system, based on what we know now, if you throw in a bunch of poly-A in a vaccine, I'd have to go through the experiments. At any rate, he was onto something.
Yeah, yep.
Happens.
Yeah, I mean, it also makes me just want to highlight how cool pox viruses are. I mean, I'm maybe a little bit preaching to the choir here to some degree, but this is, one of the things that got me really excited and interested in pox viruses biology and evolution is that you have these large DNA viruses encoding several kinases, hundreds of open reading frames that are all coming together somehow to orchestrate virus replication in really precise, at the same time potentially sloppy, at the same time just, there's a lot of really crazy, interesting biology. And by, as we mentioned sort of at the top, following what the virus does, it sort of illuminates how these ribosomes are modified or sort of organized in different ways in different species and different cells and different tissues at different stages of development. And so it's just, anyway.
You would never predict this. So another example of serendipity. And even though a pox virus, smallpox has been eradicated, people are still hammering away at pox viruses and you find new things. So it doesn't matter if we get rid of related viruses, it's still worth work and you never know what you're going to find.
Yep.
And I'm obviously I'm prejudiced, but I'm with you, Nels, and I'm really happy to see pox virology move forward. As a matter of fact, at the ASV meeting, I've been a little disappointed recently to see sort of the publications in basic pox virology seem to dwindle. But at the ASV meeting, they had 2 pox sessions, and all of them were very basic pox virology, and it was really good to see that going on.
Yeah, it's fantastic. I think pox viruses, just with the convergence of all these new tools, all these new possibilities, and the beautiful biology that these viruses conduct, I think it's going to be a growth industry. That's certainly my hope.
Yeah, you should hope, Nels, right?
For my own
that's right.
Scientific interests. Yeah, that's right. Yeah.
This episode is brought to you by Blue Apron, the #1 fresh ingredient and recipe delivery service in the country. They set the highest standards for their community of over 150 local farms, fisheries, and ranches all throughout the U.S. For less than $10 a person per meal, what they will do is deliver seasonal recipes to you along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. They ship the exact amount of each ingredient you need, so there's no waste. I love the way they send things like 2 teaspoons of soy sauce in this tiny little bottle. Just great. Their mission is to make incredible home cooking accessible to everybody. There's no weekly commitment. You get deliveries when you want them, and you can choose from an amazing array of new recipes every week, or you can let them choose for you. For July, we have, for example, creamy shrimp rolls with quick pickles and sweet potato wedges. I found out from Kathy Spindler, quick pickles, they actually send you cucumbers and vinegar, and you pickle them while the rest of your meal is cooking. Fresh basil fettuccine pasta with sweet corn and Cubanelle pepper, chili butter steaks with Parmesan potatoes and spinach. You can customize your recipes every week based on your dietary preferences, and they have a freshness guarantee which promises that every ingredient in your delivery will arrive ready to cook or they'll make it right. Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiE. That's blueapron.com/TWiE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron's a better way to cook.
I don't know if it's a coincidence, but this is always the part of the show where I get really hungry.
Must be
the same thing.
Must be. Well, here in East Coast, it's 5:30, so you're 2 hours behind, right?
True.
Yeah,
you know, Vincent, I can be hungry anytime.
Yeah, Rich has got a great appetite.
Nels, are you in Mountain Time?
That's right.
You guys are at the same time?
Yep.
No, I'm now on Pacific time.
That's right, 3 hours. Yeah. We just have 3 emails, so let's go through those before we do our picks here.
Yeah, sounds good.
Hey, Nels, the first one he wrote to you. Why don't you read it?
Okay. Tom writes, as one of those hooked on the TWiEVO podcasts and a patron, I have enjoyed every one of your shows, listening to you, Vincent, and occasional guests exploring the fascinating processes and products of evolution. As a science fan, not a scientist, often, I get lost in the specialized terminology, but I usually understand the general discussion of the interactions between the elements. My mental interpreter works kind of like closed captioning, a simultaneous line of thought summarizing what I'm hearing. In episode 20, my interpreter works kind of like closed caption. Oh, sorry, I stuttered there myself. in episode 20, this habit unexpectedly kicked in when you were talking about the intricate dance of unrelated organisms negotiating a close relationship. So we were talking about cnidarians and the algae that they associate with in symbiotic relationships. You noted that when 2 different genomes get together, initially both sides experience increased stress. There is a continuous gradation of resulting relationships from A is a parasite through A and B have balanced symbiosis to B is a parasite. The relationship can change over time, or the relationship may last a long time or may end with one rejecting or killing the other. My closed captioning channel said, he's describing marriage. I love that.
That's really great.
Play back that segment of the podcast and see if you don't agree. Tom in Austin. Tom, this is spectacular. So first of all, I mean, thank you for being a patron of the Twix podcast. We all, I know, really appreciate that. And then I really like this idea of closed captioning a lot. And so first of all, maybe let me express some sympathy for all of the jargon and terminology. I certainly have a lot of friends who aren't scientists who are at times frustrated by how if we get on to some scientific topics, it sounds like a different language. And this is, I think, will always be a challenge as we're talking about these projects and the interesting science emerging in all different corners. But I really like this idea of closed captioning, and in particular, comparing these symbiosis events to a marriage and the different outcomes that can happen there. You know, I guess I'm trying to think about closed captioning for episode 21. And this idea of mimicry. Maybe one closed captioning idea would be that imitation is the greatest form of flattery.
Right.
And that we see this sort of bubbling up at all kinds of different levels of biological organization. We talked about that a little bit, from the butterflies in the rainforest to maybe a couple of amino acids in a loop of a protein. And so, I think it's really great to think about these things on these levels, because it can just stimulate the conversation, open up new ideas, and think about this kind of work in fresh ways. So thanks, Tom.
Send us other closed captioning that comes in. It'll help us describe stuff in the future. By the way, Tom, I've had lunch with Tom.
Yeah,
he's the one. He's the one who introduced me to thread gills and chicken fried steak. So
awesome.
So this mention of symbiosis, I have to tell both of you, you'll love this because it's relevant to today's paper and also other things. On TWiEVO, we've talked about parasitoid wasps that inject their eggs into hosts like caterpillars, along with viruses to manipulate the caterpillar so they don't reject the host. We've talked about injecting into ladybugs, and the virus paralyzes the ladybug so it sits still while it protects the cocoon. Over on TWiM 155, we talked about a fly, a Drosophilid, that contains a symbiont, spiroplasma, which are sort of mycoplasma-like bacteria. They're symbiotic within the Drosophila. And when a parasitoid wasp injects its eggs into these symbiont-containing flies, the symbiont makes a ribosomal inactivating protein, a toxin that specifically inactivates the ribosome of the wasp so it can't kill the fly. It does, wow. And these toxins, they're called RIPs. They depurinate the ribosomal RNA, and they protect the bloody fly. And
so good.
This, you know about these RIPs because ricin is a RIP. That's how ricin works. It depurinates your ribosomes. Anyway, how cool. That's another symbiotic relationship where they call it a defensive symbiosis because it helps the defend against parasitoid wasps.
Very cool.
Colm writes, hi, in response to Alexandra's letter on TWiEVO 20, she may be interested in the people from a meeting I was at last week, Keystone Neuroinflammation. Includes a link to the program website. But if she wants to see the list of poster titles or attendees, please tell her to email me. And I think Alexandra had asked about something programs at Harvard, right?
Mm-hmm, indeed.
Neuroimmunology. Colm is a postdoc at UTexas.
Yeah, thanks, Colm. We'll pass that along to Alexandra for sure. And Colm has been popping up my Twitter feed with some great, really humorous responses to some of the Twitter conversations. So thank you for writing in as well. Really appreciate it.
And finally, John from Limerick writes, quick correction, Nick Lane wrote the vital question, not Mark Lane. Okay, I got that wrong. Mark Lane. That's a famous name. He used to be a lawyer that was working on the Kennedy assassination, if I'm not wrong. Mark Lane Kennedy. I think I heard him talk once, and he showed, early Kennedy assassination conspiracy theorist. Oh, he died last year. Yeah, that's not the Lane I was thinking about. Nick Lane wrote The Vital Question, which is an awesome book. Let's do some picks.
Sounds great. Yeah, I've got one. So I was inspired by, again, this idea of mimicry that we've been talking about a bunch. So I was looking back through some great videos along the way. And so, it's not only butterflies, there's mimicry happening, sort of morphological mimicry happening all over the place. And here is I think a really spectacular, gorgeous example, which is the Pharaoh cuttlefish mimicking a hermit crab. This is a video clip on YouTube from National Geographic. I think this is Nick Hubbard put this together. And so, just watch I can't do justice to the video. You have to watch the whole thing.
It's so amazing.
There's basically, it starts out with what looks sort of like maybe a hermit crab. It certainly doesn't look like a cuttlefish, but the longer you watch these cuttlefish hunting these bright blue prey fish, you realize that they really do look like little crabs. And so the idea is that the cuttlefish is changing its appearance, both its color, the way it's arranging its tentacles to look sort of like claws, and even changing its texture to look like it has the shell of a hermit crab, that this could be a couple of potential adaptive or evolutionary value to this behavior. One is that the prey fish don't realize that they're in the in the company of a predator that could reach out and eat them in less than a second. And so they just think they're around these harmless hermit crabs. Or secondly, it could be defensive as well, in the sense that there are a lot of potential predators of the cuttlefish that don't really care about hermit crabs, and so they can kind of hide in plain sight. So check out the video. I think, it's just so fun to see all of these, and just inspirational to see these examples of mimicry in nature. And this is only one of many. So I have another link to a great blog, Why Evolution is True. So this is this is Jerry Coyne, a retired evolutionary biologist from the University of Chicago. And there's a link here to different cases of mimicry in nature he's covered over the years, on his blog, and you can just find example after example of insects and other snakes and various critters that are using mimicry for evolutionary sort of advantages.
And there's also a paper associated with the Cuttlefish one. You can go look at that.
Yeah, absolutely.
Journal article.
This is amazing. Does this Cuttlefish know any other tricks, or is he, or is he just do the hermit crab? Probably just the hermit crab thing. I wonder if there's other cuttlefish that do other mimicries. It's amazing how they can change.
Yep. So certainly there are tons of cuttlefish that do certain different kinds of camouflaging behaviors where they can change their coloration or the patterns. But yeah, I up until seeing that video, I was totally unaware of this case of the Pharaoh cuttlefish. Isn't that cool? Yep.
And especially the texture of the skin or whatever you want to call it, right? It just changes it completely.
Yeah, and so the very first clip, I was intrigued, but it was really, I think, there's a few scenes, and once you get deeper into it, there's a scene or two where it's like, oh my god, maybe that is a hermit crab for a second, and it's really cool.
That's great.
How about you, Vincent? What's your pick of the week?
Mine's a little self-serving. It's our first bioarchive submission.
Oh, fantastic!
An old guy like me. But we have a paper on Enterovirus 68, which we're pretty excited about. It's work done by Amy Rosenfeld and Audrey Warren in the lab, and it's been submitted to a journal. And at the same time, Amy said, let's put it into bioarchive. So we did, and people are looking at it, which is pretty exciting. And we've talked about bioarchive before on TWiEVO, and I love it. I love the idea that your papers get up there. And here on TWiEVO, we remember Buck Tribble and the ant mutant. They put it on. They were sending it to journals. It was getting sent away without review. They put it on bioarchive, and then I don't know who ended up publishing it. Cell reviewed it right away, so maybe there's a connection. Maybe.
Yeah. That's right. How did you find the submission process to be?
Very quick. Mm-hmm. Takes just a few minutes to upload your article. They make a PDF, you check it, and then it takes about a couple of hours, 6 hours or so for them to approve it so it gets online. But it's really easy. Have you done one, Nels? Have you put one there?
Yeah, we've done several now. I think we've done 3. And it's the same and I found the process to be great. We did the same thing that you just did, which is also put it into a journal. Mm-hmm. And so our first one converted to a PLOS genetics paper. The second one converted to a Journal of Virology paper on poxviruses and some experimental evolution that we are continuing to do with them. And then the third one just converted this last week to a genome biology and evolution paper. Mm-hmm. So yeah, I've been actually really thrilled with doing this and getting the work out there a lot quicker.
Yeah, so it's great. I think this is a great mechanism, and I think people should do it. Most journals now are okay with it, right? As far as
that's right.
So
yep.
What do you got, Rich?
I have an article that was sent to me by my buddy Nassim. I have a vague feeling that we've talked about certainly this phenomenon, but I don't know about this article on TWiEVO before, but I couldn't find it as a pick. At any rate, this is an article in the British Medical Journal published at Christmas last year, 2016, under the heading Food for Thought. And it has the pun-filled title of Sniffing Out Significant P-Values, Genome-Wide Association Study of Asparagus anosmia. So some people, at least, about half of the population, will be familiar with the concept that if one eats asparagus, one's urine has a foul smell afterwards.
Count me in that. Yep.
Right? That's me as well. And anosmia is the inability to smell stuff. Okay? So some people can do this, some people can't. And there was a controversy for a long time over whether this had to do with whether or not you actually metabolized the asparagus and made the smell, or whether you simply couldn't smell it.
I see.
There are obvious experiments that you can do to determine that, and it turns out that it's all a matter of smelling. Everybody makes the smell, but some people can smell it and some can't.
Oh, I didn't know that. Cool.
And this paper is a genome-wide association study where they take a whole bunch of people that they've actually 6,000 that they've interviewed and asked them whether they have asparagus anosmia, and then looked for alleles that were or were not associated with anosmia, and come up with like a single SNP in an olfactory receptor.
Very cool.
Okay, so it all makes sense. Some people, because of differences in their olfactory sensors, can smell this, and some can't. So I have several comments on this. First of all, it's just an amusing paper with all sorts of. They actually have a little video that goes along with it where they're kind of making fun of the whole thing, and it's amusing, and it's full. The paper, of course, is full of puns and stuff. But
p-value, that's PEE.
Yeah, right, exactly.
Yeah.
So, I wonder if this variation, there must be variation in other stuff that you can smell.
Probably, yeah.
And I thought it was appropriate for this show because it makes me think about, olfaction and evolution.
Mm-hmm.
Yep.
I don't know. How would this, is there any selective advantage in having or not having this particular allele? I don't know.
It's a great question. There's certainly all kinds of really interesting evolution happening with olfactory receptors. I mean, obviously, this feeds into predator-prey relationships. And so there's very clear cases of adaptation. There's also because a lot of these olfactory receptors are 7 transmembrane spanning proteins. There is a lot of just so-called relaxed selection, where if you just have a lot of hydrophobic residues, you can kind of mix and match them without a lot of functional consequence. And so there's some well-known studies on fruit fly olfactory receptor evolution where, in some sense, it was a negative result that you could just have all of this variation with very little functional consequence. So there's, maybe a note of caution that not every one of these variants might be adaptive, but there certainly are cases where they are. So I agree, Rich. It's a really cool area of evolution and in a great amusing application of human genetics here as well, I would say.
They say that the study was conceived during a meeting attended by several of us in bucolic Sweden when it became apparent that some of us were unable to detect odor in urine after consuming new spring asparagus.
The little video has the first author and the senior author sitting at a table looking like it looks like a TV host guest discussion show, and they're sitting beside this table. The table has a bundle of asparagus sitting on it, and later in the discussion, one of the authors is making points, holding a spear of asparagus like a pointer or something like that.
Nice,
pretty entertaining.
That's great.
All right, that's TWiEVO 21. We're legal. You can find it at Apple Podcast, microbe.tv/TWiEVO. Consider subscribing. In fact, don't consider it. Do it. You have a favorite app that you listen to podcasts on your smartphone or tablet or whatever. Just subscribe to TWiEVO. And that way you get every episode, whether or not you want to listen to it. And we get a download number. Okay, so do that for us. Also think about supporting us financially. You can go to microbe.tv/contribute. We have a Patreon account. As you heard, Tom is supporting us there. And we have other ways that you can help us out as well. And send your questions and comments to TWiEVO at microbe.tv. Nels Elde is at cellvolution.org. You can find him on Twitter as Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. And hey, Rich, thank you for doing this. It's really a fun honor to cover a pox virus paper with you.
Man, thanks so much for having me. I wouldn't have missed this for anything. This is just terrific.
That was great. Rich Condit, by the way, is a emeritus professor, University of Florida at Gainesville, currently residing in Austin, Texas. You can also find him on TWiEVO pretty much every week. Thanks, Rich.
Yeah, sure enough. It's always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this show, Blue Apron. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.